type stuff, but I might lose track of time. So yeah, feel free to.
Okay. If we get to one.
Us.
Okay, thanks. Yeah. Okay, welcome everybody. If you're joining us from Facebook or in Zoom, we'll be getting started in just a moment. We'll wait till 1 o'clock though, just to make sure we're not starting too early for people. If you're waiting, please let us know where you're watching from. You can also let us know if you enjoy spiders, for example, or if you have a favorite spider. So go ahead and try out the chat functions, which we'll be using for your questions today. So I've just launched the quick poll that's already there. Oh, the zebra jumping spider. Wow. Okay. That's Nicole's favorite. Oh, yeah, I should have gone more specific. Who would name your favorite jumping spider? Okay, great. And so we've got some people that are returning to the Beaty programs, Beaty at home programs, which is fantastic in our poll. Okay, so let's go ahead and get started. I want to welcome everybody and thanks for joining us. Oops. Sorry about that. I just want to welcome everybody to the Beaty at Home. We are very lucky to have Dr. Wayne Madison here to talk to us about jumping spiders. Wayne is the director of the Spencer Entomological Collection at the Beaty Biodiversity Museum. I am Nancy, and I'm a museum interpreter, and we also have Sheila here joining us as well. And as well, you might recognize Nicole in there. So. Let's start off by finding out where the Beaty Biodiversity Museum is. I don't know if any of you have already visited or if there's anybody out there that hasn't visited the museum yet, but we are on the ancestral, traditional, and unceded territory of the Musqueam peoples. And we have this website here is a great place to learn more about their culture, about the Musqueam people and their culture. So I highly recommend their website. On the map, you can see it's the Beaty Biodiversity Museum is in the middle of UBC, University of British Columbia, and we are pretty much in the center. We're very close to things like the Biosciences Building or the Pacific Museum of the Earth and the Biodiversity Research Center. So I'll show you that in a sec. If you're coming up to the building, this is what it looks like, and you start approaching this glass atrium in the middle. You will see as we get closer a blue whale skeleton, which we're quite known for. And behind that, if you look through the glass in this photograph, you can see the Biodiversity Research Center as well. And in the center, there's over 50 researchers that are working in all sorts of very interesting biodiversity topics, such as how things work together in ecology or how things have changed over time, evolution, as well as many other things. So that building is directly connected with the museum. And so if you're going into the museum, you will see a ramp and you go down and you will see our beautiful blue whale skeleton. As you go down the ramp, you'll be underneath. And just a reminder, though, that the blue whale is just, the skeleton is just one of over 2 million specimens in the museum. So there are all sorts of things, not just the big ones, lots of tiny ones too. And they are organized in 6 different collections that have been gathering at UBC for the last 100 years. And they are gathered in cabinets, much like in other museums that store and protect their specimens. We have cabinets, but the very interesting thing about our museum, one of the very, very interesting things is that over 95% of our collections are actually in the same room that the visitors visit. And so this is a photograph of those cabinets in rows. These are the 6 different collections that make up the UBC collections, biodiversity collections here. And here's a map of those rows. So you can sort of see on this side here, you've got the blue whale skeleton, and then these rows are where all those amazing specimens are. And today we're going to focus in on the Spencer entomological collection here. And the specimens that Wayne will be talking about too are stored in this row. Okay, so we'll go ahead and encourage you to type your questions as we go along in the Zoom or the Facebook chat, and we'll read those questions out loud for you at different times. So we'll go ahead and let Wayne take it over. Thank you so much for joining us today, Wayne.
Thanks. It's great to be here. It's great to talk to you all about jumping spiders in the Beatty Museum and how we do the sort of research we do. And I'm really lucky to be at the Beatty Museum because it probably has, I'd say there's a good case that it has the best collection of jumping spiders of any museum in the whole world. And by best, I mean sort of the number of species, the representation of them and so forth. It doesn't occupy a really big chunk of space downstairs in the museum, but it's really concentrated in special things. And the jumping spiders are not a small group of organisms. They have about as many species, probably more species than there are species of birds. We're still discovering them all, but it means that it's a big chunk of biodiversity, and the museum has it really well represented. So what I'm going to do is tell you a little bit about myself and sort of what I do as a biodiversity researcher looking at jumping spiders. And then I'm really excited to answer your questions. I think that the people running the show here will be doing certain things to optimize what you see. Was there anything, Nancy, about like managing the screen? Because I'm going to be sharing screens and everything, and so it'd be good to have it so you see things big as possible.
Yes, thanks. Sorry about that. So I wanted to point out that on the upper right corner, if you're in Zoom, you should see the word speaker view as an option. And then you could hit that, and that will make sure that the person that is actually speaking will be the largest window. And the other thing that you can do as well is that you can go to that the window of the person and go to the ... And if you hit that, then there is an option to pin the video. So if you ever need to just keep the view on that one person, you can pin video by that ... menu. Thanks, Wayne.
Okay, thanks, Nancy. And I should warn you, I'm represented twice in this Zoom chat, and the one time is my cell phone that I'm going to use to do a roving thing of the little lab here in my house. So at that point, you might want to pin. Okay, so I'm going to share my screen now and tell you a little bit about how I got started and what I do. And the story, the origin story, sort of like, you know, Batman and all that stuff, the origin story of my work is that when I was 13 years old, a spider looked up at me and I was fascinated. And jumping spiders are strange because they do look up at you. They've got really good eyes. And this was 1971 on the shores of Lake Ontario. You can see here the 4 eyes up front that they look at you with when they look up. And from then, I wanted to know more about them, but not just that species that looked up at me. I think that I've just got a real drive to understand diversity, to see diversity. And so I wanted to see more and more different sorts of jumping spiders. And so the next year, when I was 14, I started drawing them. And these are the oldest drawings I have. And to me, the drawings were both recording my observations, helping me to understand the spiders by noting their features, but also sort of a celebration of how cool I thought they were. And so I kept doing that, and that became really a big part of who I was when I was first getting started as a biological illustrator. So I illustrated them, sometimes in color, and over the last many years, many decades, I've been mostly just using black and white for more technical illustrations. And so here are a couple of these. So we do these drawings as biologists in order to illustrate the little tiny bits that help us tell them apart. and also to illustrate them so that we know what their structures look like. And that might eventually be useful to understand how the structures work, how the spiders work. But besides just telling you a little bit about how I got started, I'm going to talk about spiders first, and then I'm going to continue with the idea of sort of the work that we do. So what's special about jumping spiders? Well, the first obvious thing, given the name, is that they jump. And I hope that this video works okay, streamed and everything, that it's not too jumpy, but too jerky. So here's a little jumping spider getting ready to jump. Boom. And you saw that it jumped there. They can jump pretty well. The farthest that I've ever seen is about 25, maybe a little bit more, 30 times the length of the body of the spider. Of course, a fairly small spider, which I've seen, in other words, I've seen them jump about a foot and a half. So not really long. They jump primarily to get around, as this one just did, but also to jump on prey, to jump on little flies and things. And the peculiar thing about their jumping, you might notice, is that they don't have big back legs like a grasshopper. And so they don't actually use muscles in the legs to jump. What they do is that they use the muscles in the head here to clamp down and basically restrict the volume of the head. The blood that's sitting in the head has to go somewhere, so it squirts out and it fills the legs very quickly. And so the legs suddenly snap like this because they're filled with the blood. And that snap of the legs is what propels them. So they're actually hydraulic. They do use hydraulics to jump. So the most obvious thing, though, maybe when you look at a jumping spider are these eyes, because they got really big eyes compared to most other spiders. And in fact, every one of them that you see is going to have these 4 big eyes up front. And so here's what you see if you look close. This is a rather ferocious looking jumping spider. Those are the big jaws up front. But pretty much just scary to little insects. So you see the 2 great big eyes up front. And there's 2 other little eyes just beside those. And then on the back of the head, there's another 2 eyes so that they can see behind them. And there's 2 tiny eyes in between that it's not clear what they do because it's hard to see that they do much good, but they've got 8 eyes total. So that's the way most spiders are, with 8 eyes, and jumping spiders have 8 eyes as well. But they got these 2 big ones up front. And so when you look close at a jumping spider, you can see those 8 eyes with the 4 big ones looking to the front. So what actually is inside that head? So how do the eyes work? Because there's sort of a cool story about this here. And if you were to open up the head, you'd see this. You'd see that there are these 4 big eyes up front here. I hope you can see my cursor. And then these eyes in the back. And the 2 big eyes in the middle front are called the central eyes or the main eyes. And they're different than the others, primarily because they're long. They're not round things like our eyes. The other 6 eyes, the side eyes, are more like our eyes. They're not compound, not like an insect eye. And so they, a little globular eye that sees a fairly big range of things, a field of view. But they don't see really clearly, those other eyes. But the central eyes see really, really clearly. But there's this long tube that acts sort of like a telephoto lens. So what they see really clearly. And in fact, jumping spider vision in terms of what it can resolve in terms of angles is better than a cat, which is pretty remarkable to miniaturize a little photo system that good, better than a cat, in a head that small. And here you can just watch the eyes move, because those big eyes, because they see such a narrow field of view, like telephoto lenses, they have to move to be able to see around. And so they move those big tubes side to side to scan the environment. And in fact, in most spiders, you can't see it. You don't see this happening. You'd have to somehow peer inside the head. But in this particular one that has almost completely transparent, you can see it pretty well. So this is from a cloud forest habitat in Ecuador in South America. And so here it is here, and you see those eyes moving. And so as those black tubes are moving, he's looking in different directions. Of course, he's also tilting his whole head to see what's going on. So there you go. So as I said, they hunt like cats. And that's one of the uses that they put their eyes to. They'll spot something, they'll sneak up and they'll pounce. But they also look at each other. And so here are two jumping spiders, a male on the left, a female on the right. Oh, by the way, is the video working well? Thumbs up on that? Videos are going okay? Okay.
It's great, yes, thanks.
Good. So here we see a male that's already spotted the female, and he's gone into a courtship pose. So he's about to dance for her. And he's got this weird, strange pose that he's put himself in, but she hasn't seen him yet. And so you just watch them, and she's looking around, and well, you can see the moment that she sees him, and he's starting his dance. So you can watch these spiders interact, and it's really clear that they notice each other with their eyes, and they respond. And the males will do these courtship behaviors. So I'm curious as to which ones all of you people have seen out there, and I know you could be anywhere around the world, those of you that are watching in, but I'm gonna just show you some that are around here in Vancouver, Canada, and these are some of them. There's Salticus that we find on our houses pretty commonly, the zebra spider. There's another one that's called Psittacus or Achilles. It's changing its name now. There's another one called Platycryptus. So these are the ones that we tend to see around houses. There's also, especially on the ground or in bushes, this red and black one called Phidippus, a fair bit bigger than those other ones. This one tends to scare people, although it's not really harmful. Here's another set that are on trees and bushes around Vancouver. Phanius, Metaphidippus, Pellegrina, Ivarca. And then on the ground around Vancouver, especially open sunny ground, we find these guys called Hibernatus. And Hibernatus are pretty cool because they're like little birds of paradise. They can be really colorful in the male, and the male does these really fancy courtship displays to the female, a little bit like what David Attenborough shows from New Guinea. In fact, one of the common names people use for these are the paradise spiders because of how similar they are in some ways to the birds of paradise. So just to show you a few of these paradise spiders from Vancouver, here's the male and female of one that you can find on the beaches. And here's the male doing his courtship, where he basically stands high and goes side to side, swaggering towards the female as he gets closer and closer, and then he reaches forward to touch her. Now, the squares here are 1 millimeter each. So these spiders are about 5 millimeters long, pretty small. I think if they were 1 foot long, everybody know them for how colorful they are. They'd probably be scared of them too, but they'd know them for the color. Here's a pretty charming little guy that's also in the Vancouver area. It's got sort of a rusty bum that's metallic pink in its shine. And lo and behold, when he does his courtship, he raises his bum in the air. And there you go. There's another one called Hibernatus oregonensis, and the males of this one have great big front legs, sort of like they're bodybuilders. And what they do when they do their courtship, when they see the female, is that they'll spread their legs out and they just stand there like a bodybuilder, just sort of doing Arnold in the old days at the female. They don't do much else. They just sit there and spread their big fat front legs. But the most complicated courtships are seen in this group, and there's one that lives in the Okanagan Valley in south central British Columbia. And these guys have fancy fringes on the first leg, something strange going on with the third leg, sort of bumps and colors and things, and a stripe across the face. And so when they see the female, they do this really complicated thing that if you sit and describe it, you have to describe it in about 7 or 8 or 9 different stages, in each of which all these different things happen. And I'll just show you a quick bit of it here. But he basically goes bounce, bounce, bounce. It's his third legs, his front legs. Now he's shaking the third leg on one side, the third leg on the other side. He keeps going back, third leg alternating, and then he tweaks, tweaks, tweaks. And then he's suddenly shaking everything. He raises both third legs, and he's doing these really strong vibrations on one side or the other. So it's a really complicated dance that the males do to the female. So after I got started and I got really excited about the diversity even in Canada, I became an undergraduate student and a graduate student, did my PhD. And since then, I've been a professor, first at Arizona. I was born in Ontario, in London, but I was in Arizona for a while, and then I came back to Canada, to UBC. I was lucky to come here in 2003. And in the course of that career, I've managed to go to lots of places in the world to look for jumping spider diversity. And so these are the spots of some of the places I've been, including in Asia, a lot in South America, and a lot of times in South America and Mexico, and of course a lot in North America, and once in Africa and Gabon. And when I get out to a tropical rainforest, this is what you see me do. So here we are in New Guinea, and I'm walking along, and I've got this big sheet. And this big sheet I use to catch spiders that fall when I shake or bang vegetation. And it makes them much easier to find than try to find them in and amongst the vegetation. They drop when I hit it, and I look, and they're easy to catch on the sheets. And so I'm just going there, shaking and beating the vegetation, trying to find weird little spiders. And when I do this in most tropical forests, about half or more of the species that I find are new to science. Nobody's ever seen them before. That's how poorly we know spiders. And in fact, if you look at a global scale, probably we found only about 30% of the jumping spider species that exist. We're probably still 70 or so percent unknown to us, still to be found, if we manage to find them before their habitats go. So that's me in a tropical forest. And occasionally, I also just find them hopping along the ground, or I put piles of leaf litter on my sheet and sort through and watch them scurrying away. And so in this way, I get a sampling of what's out there. When I get back down to the camp, this is a really old camera that I have. I'll get my camera out and photograph them live, because I want to record what they look like live, because in most of these species, nobody's ever done that before. And the results then of one of these trips are specimens of a few hundred species. They're pickled at this point. They're well labeled as to where and when they were found. And I bring them back to the Beatty Museum. And of course, other people have been doing this for the last couple hundred years, and other museums in other parts of the world have jumping spiders pickled in little jars. And so when it comes time to doing a study and to assess, say, the diversity in species of some particular group of spiders, I will look at what the Beatty has, but I'll also get loans from other museums. They'll send me the specimens they have, and I pull it all together and try to understand what's out there. And so that is the slideshow bit. But now what I want to do is show you, if I can. Are there any questions?
So, yeah, that's great. So I started with a question. I was wondering if there was a trip where you had the most number of new species found. Can you give us an idea of how many new species?
So you know what? It's actually really hard to tell. Hold on. It's really hard to tell, because we go get all this material and we have to figure out whether something's new, which means that we can look at it under a microscope, but then, oh, is that the same as the one that so-and-so described in 1953 from a nearby locality, or is it different? And so there's a lot of work looking through old literature to figure that out. And so there's only a few expeditions where we've really gone through everything to try to figure out what's new. Because most of the expeditions, you store it away in the museum, and it might be that the first person that actually looks at those to figure them out and study them will be in 100 years. And we expect that that's going to happen. Right. So there's only a few, in most expeditions, we actually study closely. But for one of the expeditions, in one locality that's about a 5-kilometer radius, we found about 150 species of jumping spiders And we figure probably about 60% of them are new. So right there, there's like 80 species or something like that.
Oh,
80 or
wow.
90
Yeah.
species that are new. So probably in the Beatty Museum collection right now of jumping spiders, there are about, there are probably at least 300 or 400 or 500 species new to science that are waiting to be described, documented, published. So
Was that one tropical area? Do you mind me asking if it was Papua New Guinea or another country?
no, that
or?
was Ecuador.
Okay,
The two most, by far the most diverse places I've seen are Papua New Guinea and the Amazon basin. Nothing else compares to those two. They're just amazing.
And then I've got a question from Nicole. Do you find jumping spiders in groups or usually in singles or pairs?
Oh, that's an interesting question. Almost always singles or pairs. They're not generally social, but there are some reports that nobody's really looked into that there are certain species that have aggregations and they have stridulatory organs like crickets do. And so we suspect that they talk to each other. And if it was just the males talking to the females, no big deal. But actually the babies have stridulatory organs as well. So we wonder whether those ones actually might form little social groups. But we don't know anything about them. Nobody studied them, the natural history of them. So
that's really neat. I kind of guess they might be territorial because they have their little dances for courtship. I thought maybe they could also like have a little display, but I guess not necessarily.
So the males will react to one another, and they will sort of sometimes push each other around, but there's not real clear evidence of actual territories.
Cool. And then another question from Sheila is she noticed a Ziploc bag in one photo, so she was wondering if they were being used to capture the spiders with, or
no, the Ziploc bag is just to organize the catch. So because in a single day you might walk up to this spot here in this habitat and you get a batch of vials. You'll put them into a single Ziploc bag so you know that they're all from that spot. And then in the same day, you might go to a different spot. So it's just to keep things organized.
And she's asking about the preservative as well.
It's just simple ethanol. Yeah.
And then Chloe asks, going back to the eyesight, are there any other types of spiders that have vision comparable to jumping spiders?
Not in terms of the accuracy, the resolution. There are some real specialists, but the other really strong specialists that have really amazing eyes are ones that have very sensitive eyes for nighttime. So there's some that they can't see very clearly, but they can see in very dim light. And these are like the ogre face spiders, for instance. So but there's nothing that has that really, really high resolution like a jumping spider.
Fantastic. And Rebecca asks, what would you think is your best capture of species numbers? Where and when? So I don't know if that was similar to the other question.
That might be answered. So that one site in Ecuador where we found 150 is the highest that we've ever seen in a small area like that. On a single day, I've never counted. I don't know. So.
And Sheila asks another question: is it possible to record their sounds?
Yes. There are. If you try to use old-fashioned things like microphones, it's really hard because they're very, very faint. So the standard way that people do it nowadays is they use laser interferometry. So they shine a laser beam at the ground where the spider is, and they get the reflection. And by using the interference of the light beam, they can see that the substrate is shifting, and they can convert that to sound. So they can actually record using a laser beam, and that's much more sensitive, and that's the standard way that people do it nowadays.
And I have a different question. Do the females usually eat the males after mating, or unsuccessful suitors?
No, it's in any of the observations I've made, it's quite rare. It does happen occasionally. Sometimes sometimes people that are studying them notice it. They see it somewhat frequently because we're studying them in an enclosed space, which gives the males less opportunity to run away. So sometimes that makes it harder for the male, but generally the males are really good at getting away if the female starts to be aggressive. So.
Okay. That's interesting because I thought maybe that was a driver for the really elaborate courtship dances, is maybe that they had to make sure the female didn't see him as food, but I guess.
No, it could be. It could be, but it would only take a 2% risk or something to make that still a valuable thing to do.
Right. Also, we've got a question from Rebecca. Have you seen a difference in biodiversity number of species in a single place after some years? So I guess being able to see a difference after coming back to a place.
So I can't say that I've done enough time cross sections to know that. Like a lot of these places, I've only been there once. But what I do notice is I look at some of the collections I made when I was first getting started in the 1970s, and I look at the vial and I think, how did I find 100 spiders in a day? So, because it's useful for us sometimes to have a big sample, because that way we can look at variability, and that'll help us to understand the species. So sometimes we will collect 100 in a day, especially if the population has tens and tens of thousands of individuals in it, right? And I looked and I said, how could I have ever seen 100 in a day? You don't see that anymore. I've not seen them. Like I remember abundances when I was younger, that I've not seen those abundances since. So it seems to me, but it's not a real study, right? As a scientist, I would like to have that a little better documented, but my impression is that I'm not seeing them in the same abundance I used to when I'm getting to comparable habitats.
Thank you. And Jeff, we'll just ask Jeff's question and we'll let you show us your other screen. What animals prey on these spiders?
Oh, well, so there's predation by lizards, by birds, and also by other jumping spiders. There are actually some jumping spiders that make a living eating jumping spiders. Not the same species, but other ones. Now you might think, is that cannibalism? Well, not really. Given that the group has as many species as mammals or birds, it's sort of the equivalent of a tiger eating a mouse, right? So yeah, they'll eat other jumping spiders. And in addition to that, there are wasps that specialize in going after spiders. So you'll see these little wasps going around the ground that have sort of bluish wings and they flick them quickly, sort of dark black blue. And that thing's looking for spiders. And so they'll sting the spider to paralyze it and put an egg on it and pack it away and leave it behind. And that little larval wasp will gradually eat the paralyzed spider, avoiding the most critical organs until the very last minute, and then it'll eat the critical organs, and then it'll be, big enough to pop out and become an adult wasp.
Great. Should we? We'll ask Sheila's question too then. Have you ever collected in Borneo, known as a very biodiverse country?
Yep, I have. I've been lucky enough to collect once in Borneo on the Sarawak side, and so in the Malaysian side. And it was quite impressive. Still not as diverse as New Guinea or Ecuador. And possibly one of the reasons for that is that my understanding is that the forest diversity is different there. It's mostly dominated by one family of trees, whereas in Ecuador, for instance, it's quite a diverse set of families of trees that make the forest. But for whatever reason, Borneo is impressive, but not as impressive in terms of spider diversity as Ecuador or New Guinea. Now, maybe that's something quirky about jumping spiders, but my understanding is that's somewhat of a common pattern in other groups as well.
I think that's,
Oh, there was one?
We'll go ahead, yeah.
Okay.
One more. How many species did you find of Salticidae in Mexico? Do you consider this a high species diversity?
Yeah, so Mexico is really interesting because it goes all the way from neotropical rainforest in the south, say in Chiapas, Yucatan, all the way up to Veracruz. But it also has Nearctic fauna, so it gets all the way up to the fur zone, the Canadian zone in the mountains. So there's when you're collecting in Mexico, you can feel sometimes like you're in South America and feel sometimes like you're in central or northern Canada, depending upon your elevation. But the most remarkable thing in jumping spiders about Mexico are all the mountains. And so all these mountain ranges act a little bit like a Galapagos Islands in terms of isolated patches that allow diversification. And so compared to Ecuador at any given site, it's not that diverse. Like it's not anywhere near like Ecuador. Over the whole range of Mexico, though, when you look at how many species there are in different places, right now I think there's 200 species recorded more or less from Mexico of jumping spiders. My guess is that there's at least 1,000. And it is an amazing place in terms of these things in the mountains. There is one group that we've been studying recently that has about 5 species described. And on a single trip to Mexico in 2017, we found about 50 species. And there were only 5 described before we started, right? And so it was like, that was a single trip. Mexico is just incredible in terms of the montane biodiversity. It really is quite impressive. And in fact, the way to think about it is that the fauna, the jumping spider fauna of the United States and Canada, the home of it is Mexico. And what we see in the United States and Canada is just sort of this halo extending north from where most of the diversity is, which is in Mexico itself. So, yeah, cool place.
Thank you.
Okay, I'm gonna try to show you a little bit about the spiders themselves, sort of, or my lab, I should say. And so you might see, oh, I know what I'm gonna do. I'm gonna mute. Okay, oh, just a minute. Just setting up here. Okay, so I think if this works, you will be able to, you can see, can you guys follow me around here?
Yeah,
Yeah,
it looks
Wayne,
good. Looks good.
Okay, so I've got 2 microscopes at home because of COVID. I'm really glad that I could bring my research home. And this is the basic dissecting scope where I just look at the whole bodies and sort of sort things out. And this is the scope in which I do the drawings. And so I'm going to show you one guy here. And I hope this works because I don't really have it set up properly. Oh, there we go. Okay, let me see if I can get this into the, so this is a little guy from Borneo, it turns out. Sarawak Lambir Hills National Park. It's hard for me to see what. So this is a jumping spider that is fairly long and thin, and it lives on big palm leaves in the forest. And when I was out there, I found this thing and I thought, well, I don't remember, I didn't ever see anything quite like it before. And when you look in the literature, you don't find anything quite like it either. And so there he is. And it turns out that after study, it's become clear that it is a species new to science. So this particular specimen, we've chosen to be the reference specimen for the species name that we're going to give it. And the species name we're going to give it is, don't publicize this, please, because you're not supposed to publicize species names before they're officially published. This is Pedilothorax badut. Badut is the Malay word for clown, because we used to call these little guys the banded clown, because they walked in strange ways. So there, he's right there. And because he's going to be the reference specimen, in his little vial, where he lives in the museum, we're putting a red top on it. So the red top means that's the reference specimen. At least that's the tradition in spiders. And so what I did a while back is that I went to this scope and I plucked out a little bit of him that is one of our key things that we look at with spiders. It's a structure that they use in mating. And I use this microscope in a way that's not typical in the sense that the light's coming from above rather than from below. I put the little bit on a slide there, and then I can look at it. But it turns out that this is a special apparatus here that helps me draw it. Because, and this unfortunately I wasn't able to sort out for you guys today, but when I've got this all set up, when I look in here, I see two things. I see the specimen there, but at the same time, overlaying on top of that, I see whatever's there. So if I put a piece of paper there, down there, and I'm looking through here, I can see the piece of paper and I can see the specimen, which means that if I take a pencil here, I can actually trace the outline of what I'm seeing. So this is called a drawing tube, and it's sort of a way to cheat to get all the proportions right, because I'm actually tracing the image as I go. And so the result of this was these drawings here that you can see that I've done. And you might wonder, oh, by the way, is the audio coming okay here too?
Yeah, it's great. Yeah, we can see everything too.
Good. So these drawings that I did, that drawing there is actually the bit of that specimen that I showed you before. And so this will be in a publication that we're putting together to describe this as a new species. And you might wonder, in 2020, why are we using good old-fashioned hand drawings? Why don't we just take digital photos and use Photoshop? Well, there are some people that are doing that, but it turns out that there's still value in the old-fashioned drawings, partly because with a drawing, you can basically focus in and highlight the details that you've noticed after a lot of study, and those details might not show up on any single photograph. So one way I'd like to think about it is that a photograph is a little bit like data, and a drawing is a little bit like a theory, because I'm able to show my understanding of the structures in a way that might not come across clearly in a photograph. So I'm gonna now go back away from this.
Oh, and we had a. Whoop.
Oops, that's me.
Okay.
Okay. So, okay, you know what? I'm gonna quit. Just a minute. Come on.
Okay, I'll just ask Sheila's question while you're setting that up. But she was just wondering what magnification level you have. What magnification level you have on your dissecting? Okay.
Sorry, I didn't hear that because I was just
sorry.
Okay, try again.
Okay, what magnification level do you have on your dissecting scope? Sheila was wondering.
So the dissecting scope only goes to 90. And in fact, that's one of the reasons that I use that compound scope. I can go much higher and go to 200 easily. And also compound microscopes just have much better optics typically. So I can see lots of tiny things.
Thanks. And then Nicole says the drawing is so much more clear than the photos, and it's a neat way to show consensus of a structure. Yeah, I thought that was a really great description of that drawing versus photo. Yeah, thank you. Anybody else? Not right now. Okay, no more questions right now.
So what we do then is that and I'm going to show you, do some screen sharing here. Of course, I've looked at these things, I've done some drawings, but we also got to compare them to the literature. And so here, for instance, is one of the bits of literature I've been looking at recently, not to compare it to that one species, but some other species. This is published by Eugène Simon in 1901, and he used to write his descriptions in Latin. So I'm struggling right now to figure out what in the world he's saying about the spider, because I don't know Latin very well. I can use the bits of Latin that I've learned as a biologist and the fact that I speak Spanish to sort of figure it out somehow. But it turns out that in this work of discovering species, we often refer to papers that were published 100, or 200 years ago, because those old papers, if they were dealing with a species that nobody's seen since, there are one reference points and they stay valid in some way or another. So this one, for instance, this Pedilothorax, nobody's knowingly seen a specimen since 1901. And so we think that somebody may have found it recently. And so now we're going back to this old literature to compare to see if this is in fact the thing found was Pedilothorax. But at the same time, we also then look to newer literature. And so here's another one that is a newer paper done by a group of biologists in India. And interestingly enough, they published this in a Russian journal, so that there's both English as well as certain things in Cyrillic and a little Russian abstract. But in this paper, they described a new genus from India and some new species that they found within that genus. So there is literature from people from all over the world, and it's both old and new, all in this long-term effort by biologists to discover the biodiversity that's around. And so what I've ended up doing for this project is, as I said, I did the drawing and then I composed it into this plate. So here's the plate describing that we'll use in the description of this new species. This is the drawing that I showed you right there. These are drawings of female bits, and then below it are photographs that we took of the species when these specimens were alive. So here's the male on the left, and there's the female on the right. And when we were out there in the field in Borneo, in Sarawak, we called them the banded clowns because they looked slightly silly with these funny white bands and spots, but also they walked in really strange ways. They were constantly waving these front legs, constantly waving the front legs. And we have no idea what this means. It's as if they're either trying to disrupt their outline so predators don't see them, or they're trying to look like something else. It's hard to know. And by try, I mean, not intentionally those spiders, but those ancestors of theirs that have done this succeeded better than the ones that didn't because of some role in the ecosystem that this set of colors might play. So we put together illustrations like this, and then we write an accompanying description and publish it, and then this thing will have the new name. And actually, that's not the new name that it'll have, because I've discovered since I put this plate together that things changed and we understand better what the relationships are, and this will be a Pedilothorax, not a Baboa. So that's basically an outline of how we go from finding things in the field, studying them, looking to old literature and comparing to species that are already described, and writing up a publication of a new species. And one of the issues that we face is that given that there are about 6,000 species of jumping spiders described, there are probably at least 10,000 more out there yet to be found. And there's only a handful of us working on this family. Just, a half a dozen of us working actively around the world. Starting to be more, we're starting to get our numbers up. I would say in 5 or 10 years, we'll be up to maybe as many as 20 people actively working around the world. But it still means that there's an awful lot to do. And we're only just chipping away at it. But there's some really cool spiders out there waiting to be discovered. And that is all that I had in terms of the things for the show and tell. And I'm quite happy to answer questions and so forth.
Okay, great. Thank you so much, Wayne. We've got Rebecca asking, I think I'm seeing that she's asking about the importance of morphological structures versus DNA genetic analysis for species identification.
So I guess they're both useful. I guess the way I see it is that when we have only a handful of people working on this great big family and we need to move quickly, whatever will get us, whatever data will come the fastest is probably the highest priority initially, because we need to move quickly. And so morphology does a pretty good job. It does well enough that if we focus on the morphology, I think we can move a lot faster than we can right now with the genetic data. Because really, to make the genetic data to be better than the morphology, we need quite a few different genetic loci, which is a little more expensive, takes more time, and so forth. So in my view, the best thing to do is to work quickly with morphology, and then for particular problem cases where we really want to start to get into more detail, then we can get a lot of genetic data. And if we get that genetic data, we'll be able to do a better job of understanding species. But I think it's not worth it to get to that, you know, fifth digit of accuracy if we haven't even got to the second digit of accuracy, in a sense, by even discovering the species in general terms first. So I tend to go morphology first and worry about genetics when I want to get more precise. But precision at the moment, being a jumping spider person, is something we can't afford that much in most cases.
Thank you. Rebecca, feel free to follow up if you were asking about anything more about that. My thought too was, I was wondering, when you've put together a phylogeny of the jumping spiders, and I wondered how many of those species then have been analyzed genetically.
Oh, good question. Actually, you just reminded me that there was something else I was going to show you guys.
Oh yeah, for sure.
Good for that. So in that group that has the banded clown, I'm just going to show you the way that we're sort of doing this these days is to get typically now for the phylogeny, so the evolutionary tree, putting the spiders on an evolutionary tree, the best data is the genetic data. And actually that's the fastest data, in fact, because it takes a long time to get enough morphology to do the tree in detail. So this is genetic data from about 1,000 genes using one of these new sequence captured genomic techniques, which isn't too expensive and gets a lot of data. And this is about the group that I showed you, the banded clown, and its relatives. And so starting from these data, and you'll notice these are gene sequences, A, C, G, T, and all that, and you'll see similarities and differences. There's some differences. And the methods that we use to analyze this consider the similarities and differences among these species in the genetic data, and they try to reconstruct the evolutionary tree. And they don't simply use the similarities and differences in a raw way. They consider it in terms of how evolution tends to work and a particular model of that. And so it's this fancy statistical thing that they do. And this is what popped out. And it turns out that when this popped out with the genetic data, it was like, oh, this makes a ton of sense. And it makes a ton of sense in terms of what we know about the morphology. And that's typically the pattern we see. When we get this lots of genetic data, it typically shows us an evolutionary tree that makes a ton of sense to us, knowing the morphology. So there's a lot of signal in the organisms for their history. That signal is seen, signal for history, who's related to whom, is seen in the genetics, but it's also concordant to what's seen in the morphology. So in this particular case, for instance, in these guys here, I'll just sort of, these guys here, for instance, these guys here, they all have a long thing somewhere that these other guys have a short thing. And these ones here are all sort of flat. These ones here are sort of big and chunky. These ones here are sort of long and delicate. These ones here have funny little ridges on the side of the jaws. And in fact, before we did the genetic data, no one had really studied the morphology really closely. And so it turned out that the genetic data really helped us to see the morphology properly. But now that we've got this, we can now go identify them using morphology for the most part. Like if you hand me one now, based upon this, I can say, oh, that's an Indocadilla. Oh, that's a Paranthus. Oh, that's a Bavia. It's really clear now what the different groups are. So, but this goes, you can see that this sort of biodiversity research, discovering species and putting them on the evolutionary tree, goes all the way from boots in the mud in the rainforest to doing drawings that could have been done 100 years ago, to doing high-tech genomic molecular work with fancy statistical analyses. It's a really broad spectrum of approaches that we take to sort of get this comprehensive view of the different species and how they connect. Nancy, I don't think I asked you, and I think that was a long thing, didn't quite answer your question.
No, that was great. Thank you. I think Sheila has a question. We can go to her.
Yeah, Sheila.
I do. I know a certain amount of tissue is generally necessary for the DNA analysis. Does one spider suffice for that material? I don't know, weight-wise, volume-wise, or
Yeah.
do you find that you need more than one specimen of one species?
So it depends on how much money you're willing to put into it.
Oh.
So if you think about the work to get a sequence of the genome of Neanderthals or Denisovans, like from fossils, those studies that looked into our humans' nearest relatives, you have to have incredibly good techniques to pick up the tiny bits of DNA that are still in a fossil that's 50,000 years old. And so if you're willing to put that money into it, you can have a teeny, teeny, tiny bit, right? But if you use the sort of basic techniques that people use, which is what we use, we don't put that much money into it. For these spiders, the one that I showed you under the microscope, it's good enough to have about 2 of its legs.
Oh.
So 2 of its 8 legs. That'll do it.
Mm. And you're just grinding that up.
Yep. Yep.
Okay, cool. Thanks. Yeah. Yeah, go for it, Nancy.
Oh, I was just going to mention a comment from Facebook, and we've had some other comments as well earlier about how cute the spiders were and how interesting they were, but also that somebody else had a microscope at home as well, so they were relating to that. And also we had a question about capitalization here. I'm sorry, I missed the thread on it, but why are some capitalized is a question, and they're meaning the CGAI stuff. Do you know what they mean by that, Wayne?
So Facebook might have to, Lisa on Facebook might have to clarify this. So if you're talking about these, I think you might be talking about these. I'll just show you something here.
It was the genetic.
Yeah. So that's
yes.
because it's actually, there's a very specific reason that was done. And it was that if you look at these things, these are include species that are well known, like Aculus floricola here, Colonus hesperus. Those, they don't have the name of the species capitalized, whereas these ones have these capitalizations in them, and that's because when we first found them, we didn't know what they were. So those are actually just code names that we use for them, and we put them in capitals and make them seem weird, just so that nobody mistakes them for a real name. So they're just our internal code names for the species before we figured out what they are. So that's all. They won't get published like this in the outside world. So this file that I'm showing you, I probably shouldn't be showing you just in case weird things happen with it online. But it shows how we refer to things internally before we're ready to publish. So it's our own little pet name study.
There might have been something else she was looking at before the tree was up, the rainbow genetics.
Oh, the capitalization of the data. Yeah, basically, there are so many little conventions in this field, right? If the A, C, G, and T, the nucleotides are capitalized, it means that the original information that led to that being interpreted as the sequence was better information. And if it's lowercase, then it's a little bit dicey that that's actually an A. It might be a C. It's hard to tell. So it's just an issue of certainty.
Oh, nice observation.
Yeah,
Lisa.
sharp eye.
All right. I don't see any others, Sheila. Do you see anything else?
No, not yet.
Oh, there's so much we could ask. Okay. Good. Okay, well, the timing is actually really perfect. So I think I want to thank Wayne so much for sharing all of that with us. I'm sure we all learned something from the session. I'm just going to share my slides here just to say goodbye, but thanks again, Wayne.
Thank you. If I may ask, I'm just curious, do you know where people are from? If
Oh, yes, please. Everyone, go ahead and type it in the chat.
I'm curious, where are people from?
Okay.
Do you want to put it in the chat there?
Yeah, Sheila, why don't you just tell us while you see people typing in?
Yep. So we have someone from Vancouver, someone from Nevada, USA,
Oh,
Port Moody, another one from Vancouver saying fantastic talk. Thank you. That's 4. Someone from Burnaby. And
Rebecca, ¿es de México?
is Rebecca still there?
Yep, she's still there listed.
Yes, yeah.
she did ask about the Mexico biodiversity. So there's an interest there for sure in
Like the
Mexico.
farthest that we've got.
Anyone else want to contribute their location just for understanding the breadth of interest on jumping spiders?
Well, yeah, and thank you for coming. I guess so, Nancy, you were going to say a few more things?
Yeah. Do you have any last thoughts you want us to share? And then I'll just say goodbye after that.
Well, yeah, maybe I'll just say that, you know, just pay attention to jumping spiders. They're fun to watch, and probably wherever you are, there's going to be a batch of species. And probably the best way to see diversity is to go out to look in different habitats. And by that, I mean, go look on leaves, go look on tree trunks. Most people tend to see just the species that live on houses. And you don't have to go too far to see some really different ones. So go to an open sandy place on the ground on rocks or something like that. And just remember that to a spider, a habitat is tiny. So they're small. So when you think about, oh, this is a forest, this is a habitat, what they see is 100 different habitats. They see, oh, there's the side of these trees, there's the side of other trees, there's this sort of leaf, there's that sort of leaf, there's this sort of leaf litter, there's that sort of rock. So there are lots of different habitats for a spider in any given thing that we see as a habitat, which means that there's probably a lot of different species there. So just pay attention to the diversity of habitats and pay attention to the diversity of the spiders, and just enjoy, have fun.
That's
If
great.
if I could add something in there very quickly, Wayne, since I've been, my world has been open to jumping spiders. Every time I see a spider, I sit and watch to see if it's going to jump. So that's been an addition to my
Cool.
outdoors activity.
Welcome.
And just another one from Vancouver saying, thank you, this was fascinating. Oh, one more from Facebook. I'm in the Okanagan Valley and totally enjoyed this as I'm interested in various insects and especially jumping spiders. I keep my eyes open every time I leave the house. Fantastic.
Yeah.
Oh, that's
The Okanagan's
great.
got some great ones.
That's great. And then, of course, we'll be posting the recording as well, and people will be able to watch even if they weren't able to join in right this moment. So thank you again, Wayne. If you're looking for a way to support the museum, we've got a couple of websites or sites there, the donation page and the membership page. And just wanted to let you know that next week, same time, same day, Wednesday, 1 p.m., we're going to have another Beaty at Home, and it'll be about the Discovery Lab with our specimens. So we're very excited about that because it'll be the interpreter's first Beaty at Home back in the museum building. So Kashyap will be leading that. So please join us next week for that. And if you do have any feedback about any of these, please do go ahead and fill out our Beaty survey. We'd love to hear your input. Thank you. All right, thanks everybody. Feel free to leave the meeting or the stop the video. Bye-bye. Thanks.
Thank you, Wayne. Wonderful.